Okay, good morning, everybody. Thank you for joining us as usual. Hope everybody's having a good week and a good start to your day. As usual, we'll just give it a minute to get people in as attendees. We have a really great lineup today of presenters, and it's covering a very important topic. So we're really excited to hear from our presenters today. Before we get started, as usual, we have a couple updates. Dr. Song Cheng has, of course, been running occupational health for us. Song, do you have any just brief updates for us?
Yeah, sure. I thought I would mention this week something about the issue of asymptomatic screening. I know that there have been a variety of different approaches between the broader university versus the health system and the medical school in terms of how we're approaching asymptomatic screening. You know, the differences have to do with the fact that we're a healthcare environment and both we're essential workers, but also that our experience over the last 6 months has shown that our PPE program is incredibly effective and that the overall rate of transmission in our environment has been exceedingly low, as I've shared often. That said, we are also aware of the benefit of surveillance for community reassurance, for reassuring our healthcare workforce, reassuring the community that we continue to be a very safe environment both to work and to receive care. So we are launching a couple of pilots of asymptomatic screening. You recall we had done this several months ago originally, and found very, very, very low rates, around 0.5% positivity. So we are now going to start pilots where we are using technologies, services that our laboratory colleagues have developed. We'll be doing mid-turbinate swabs, self-administered swabs, and pooled testing specimens, and both of those technologies will allow us to do mass screening on a much more cost-effective and lower resource intense basis. So we are going through a variety of clinical areas, and we will be rolling this out. We've already launched it in a couple of units. We've done over 300 tests so far, self-administered and pooled, and we had so far 0 positives. But I think this is a way that we can, in a reasonable, rational, cost-effective manner, continue to reassure our community internally and our patients that we are a safe place to work and receive care.
Dr. Cheng, thanks so much for your leadership in this area, and great to hear such the relatively low rates of positives. So thanks so much, and Dr. Cheng will continue to keep us updated, and of course can answer questions as needed. Dr. Huja, I've been running through meetings on service and now joining us to present some updates as usual. Thanks for joining us.
Thank you, and good morning, everyone. I'll share my screen. So this morning, as we get started, we have a total of I'll start actually with the overall trend. Across the country, you can see that the new cases are decreasing, and that's been a steady decline, fortunately. Within our own area, while I'll show you our inpatient numbers, we have seen that the numbers are decreasing, but from that peak that we had in late July, a little uptick and then down again. There is quite a bit of discussion within our hospital leadership that 1 or 2 weeks after Labor Day weekend, the numbers may uptick again, similar to the trends that we saw after other major holidays. So we'll be prepared for that. This morning, we have 27 COVID patients, of which 9 are in the ICU and 6 are intubated. When we look at our total numbers from March 1st till August 30th, there have been 396 are at SHC. From those SHC numbers, the ICU numbers are slightly over 20%, and that, is increasing very slowly, and we'll just have to watch the trends. It's not a dramatic increase. And same with the number of deaths, just a slight uptick from the stable 5% we had seen over time. And these are deaths just at SHC, not Valley Care. The female population just slightly over by a few percentage points than the male. And you can see the age distribution remains pretty much the same as what it has been for the past few months. Just breaking down the deaths by both age and gender, you can see, unfortunately, there are more in males as is acknowledged across the globe, and the breakdown that you see here by age. And then finally, just want to thank my data team, and thank you, Errol. I'm going to head off to rounds. Stay safe, everyone.
Thanks so much, Dr. Huja. Take care of yourself. Okay, great. Well, we will move on to our presenters, but before so, I also wanted to just mention, as usual, our upcoming presenters next week. We have Dr. Karina Doe, who's going to give us an update on vaccines as well from her perspective, and she's joining us again for Grand Rounds to keep us updated on that topic. The week after that, we have the approach to monoclonal antibodies. The Vice President of Regeneron, David Wenrich, is going to present on that. And the week after that, we have our former FDA Commissioner, Dr. Robert Khalif, who's going to have a conversation with us and talk and answer your questions as well. We have this week two wonderful presenters, Dr. Taia Wang and Dr. Bali Pulendran, who's joining us again for Grand Rounds. Dr. Wang is the Assistant Professor of Medicine, Infectious Disease, and Microbiology and Immunology. She's finished her PhD in Mount Sinai in virology, an MD in Mount Sinai as well in medicine, of course. And she did a Master's of Clinical Investigation at Rockefeller Institute, followed by a postdoc training at Rockefeller University, focusing on Fc receptor biology and human immunology. Dr. Wang's laboratory uses in vivo and in vitro systems to study how antibodies can modulate viral infections or antiviral vaccine responses through signaling interactions with the Fc gamma receptors. She's won a number of honors and awards, including the Searle Scholars Award, which is a pretty large research grant award. And she's part of the Zhang Zuckerberg BioHub Clinical Investigators. So we're very happy to have her join us today. Along with Dr. Wang, we have Dr. Bali Pulendran, who, as I mentioned, has joined us earlier this year. You may have seen him present for Grand Rounds earlier, talking about part of our vaccine update in immunology. He's the Viola L. Horton Professor of Microbiology and Immunology. He received his PhD in Immunology from the Walter and Ezra Hall Institute at the University of Melbourne, Victoria in Australia, and did postdoctoral training at Immunex Corporation in Seattle, Washington. His research focuses on understanding the mechanisms by which innate immune system regulates adaptive immunity and harnessing such mechanisms in the design of novel vaccines against quote unquote global pandemics. Dr. Pulendran, as I finish here, I'll go ahead and ask you to share your screen.
Yeah, sure.
Thank you. Sharing mine. And it's also noted he's begun to apply systems of biological approaches to predicting the efficacy of vaccines. Dr. Pulendran's research is published in all sorts of major journals as well, Nature, Science, Cell, Nature Medicine, and so on. He's had multiple grants from NIH and also grants from Bill and Melinda Gates Foundation. As you can suspect, both of our presenters today are extremely experienced in this field they're going to talk about, and I'm extremely happy and lucky to welcome you both. So Dr. Wang, Dr. Pulendran, thank you so much for joining us today. Dr. Pulendran, you're going to go first, so thanks for presenting, and we'll try to leave enough time for Q&A after.
Okay, good. Thank you very much, Errol. I'm really honored to be presenting at this Grand Rounds together with my good friend and colleague, Taia Wang. Can you see my screen?
Yes, looks good.
Very good. Okay, so today, Taia and I would like to discuss a topic of extreme importance as we grapple with this global pandemic. And that is the issue of how our human immune system responds to this virus, and to what extent we could harness the power of the human immune system to control this pandemic. Of course, this is a topic of great relevance, probably the highest relevance from a global health perspective today. And the approach we have taken in our lab is what we call systems immunology, because the immune system is a system. And so we feel that to understand the immune response to a virus like SARS-CoV-2, we need to understand it at multiple levels of organization of the system. The alternative title for our talk is Warp Speed Immunology. We've all heard about warp speed vaccines, and we feel that warp speed immunology is an appropriate term. Let me explain. So I had heard about these cases coming out of Wuhan, China, sometime over the Christmas holidays. And it just didn't register because one hears about these epidemics that come and go. And of course, we're very interested in how viruses interact with the immune system, but we just didn't feel that this could become a pandemic. But still, I started talking with some of my friends, including Professor Nadine Rupail, who's a wonderful infectious disease clinician who works at Emory. And she told me that this was in January, February, that yes, in Atlanta, they had some cases that they were seeing at the hospital. So then I reached out to another friend of mine, Professor Malik Pierce, who is a world-renowned virologist who is at Hong Kong University. And he was the man who first isolated the first SARS virus back in 2003. He became very famous at that time. So I spoke to him and said, Malik, what's going on in Hong Kong? And he said, oh boy, we're seeing a lot of cases in Hong Kong. And so I said, oh, that's interesting. So then the next day I spoke to our biosafety office folks at Stanford, Ellen Siegel and Ryan McAllister. And I said, we'd like to start working on SARS-CoV-2. Is it possible? So thus began a dialogue. They said, you need to talk to the CDC, get CDC import permit to get this virus, to get samples from Hong Kong, which we did. And within a period of, I would say, 3 weeks, we had the CDC import permit from the CDC. Also, it was tremendous. I was so grateful to the warp speed with which the biosafety office worked with us and their level of engagement, but also the level of engagement at many levels of administration at Stanford in which they made this possible. I'm truly grateful for that. So 10th of April was a great day for us because some 300 samples arrived by World Courier from Hong Kong. That was one of the highlights for us. We were babysitting World Courier, the website, tracking the package. So that was the 10th of April. On the 4th of May, so that's about 3 weeks, we submitted the paper to Science. And apparently the reviews must have gone off on a summer holiday or something because they took a while. But the paper was accepted on the 4th of August and published on the 11th of August. So it was a warp speed immunology exercise. So who did this work? The people who did this work are shown here. Really, the man who led this work is this young postdoctoral fellow, Prabhu Arunachalam. He was just an amazing force in terms of leading this effort, ably helped by Florian Wimmers and Thomas Hagen in my lab, working collaboratively hand in hand with Madeline Scott in Purvesh Khartri's lab and various people at Holden Makers HIMC. So this was our COVID-19 task force, if you will. So that's the paper that appeared just a couple of weeks ago. And I'd like to tell you about this. So what were our goals here? Our goals was to probe the human immune response to SARS-CoV-2. As we all know, infection with this virus can result in many diverse clinical outcomes, ranging from asymptomatic disease, mild, moderate disease, severe disease, to critically ill patients. So what we wanted to do was to map out the immune response in these various clinical outcomes and see if we can discern any insights about what's gone wrong in people who don't progress very well. So what we did was to collect blood samples. And of course, being at Stanford, we're so lucky in terms of the array of technologies that we have in terms of analyzing any aspect of a biological system. So we were able to analyze the cellular changes that were happening using CyTOF, the changes in the expression of genes in response to this virus using RNA-seq, the changes in cytokines in the plasma using Olink, changes in metabolites, as well as the antibody response. And our questions were quite straightforward because, of course, on the one hand, the virus had been discovered, isolated, sequenced, and vaccine efforts were well underway. But on the other hand, the host immune response to this virus back then, when I say back then, it's only back in March, was a black box, effectively essentially a black box. So what is the nature of the immune response to COVID-19? Is there an immune signature that can predict disease severity? And what is the immune response in asymptomatic individuals? So we started with the CyTOF analysis together with Holden Maker and Natalia Siegel at the HIMC, in which we analyzed 40 samples from Atlanta and 100 samples from Hong Kong. And to cut a long story short, this is a little bit technical for people who are not immunologists, but I'll talk you through it. This pretty looking picture here is what's called a UMAP. Essentially, that's looking at all the different cell types in a drop of blood. And you can classify these different cell types as shown here in the legend. And this is what a COVID patient in Atlanta would look like. And if you distill this into a few simple histograms, you can see that in a COVID-infected patient, the frequencies of this cell type called plasmablasts. These are the cells that Taia is going to tell you about. These are the cells that produce antibodies. So massive increase in the frequency of these plasmablasts, as well as these other kind of immune cells called T cells. These are the T cells that the CD8 T cells are commonly known as killer T cells because they can kill virally infected cells. So that's what it looked like from Atlanta. Now you have to remember at that time, we were seeing cases coming up in China and Hong Kong and starting to see cases in America. But one of the questions was, are the responses in these different populations similar, or are there differences in different populations? So to that extent, we were lucky, as I said, to have samples from Hong Kong, in which we also analyzed the same, performed the same analysis. Effectively, at this high level, you can see the similar sort of response in both Atlanta and Hong Kong as well. Another interesting aspect of this analysis was that the frequency of this cell type, it's called PDC, plasmacytoid dendritic cell. It's just an obscure cell type. Many of you may not have heard of it, but it turns out to be exceedingly important for viruses, because when you have a virus that goes inside the body, this is the cell that fights that infection. It produces a cytokine called interferons. Interferons, as many of you know, are critically important in controlling viral infection. And interferon alpha, this is the cell type that is dedicated for production of interferon alpha. And so we found that this cell type was diminished in its frequencies in the blood. So that was interesting. But not only that, what we saw was that this cell type was crippled. When I say crippled, the ability of the cell to respond and do its job that it's evolved to do was severely impaired. And we got the first hint of this when we looked at this molecule mTOR, mammalian target of rapamycin, in the CyTOF analysis, was greatly reduced in the cell type in both cohorts. And we suspected that this may imply that this cell was unable to produce interferons. And we did so because of our previous work that we had published many years ago, showing that in the cell type of plasmacytoid disease, this pathway of mTOR was the pathway that mediated interferon production. So indeed, it turns out that this cell type was crippled in interferon production. And you can see this here very clearly. It's a very simple experiment. You take a drop of blood from a healthy person like you and I, or from an infected person, and you isolate the blood cells. And then in tissue culture, in a test tube, you stimulate the blood cells and say, under the best of stimuli, can these blood cells from a healthy person produce interferons, this antiviral molecule? Of course, based on previous work from many other labs, the answer is yes, indeed, they can make interferon alpha, and you can see lots and lots of interferon alpha. You can also see TNF alpha. That's what a healthy person looks like. But we asked the same question, but in a COVID-infected patient, and you can see that there's a substantial impairment in the capacity of those cells to make those antiviral factors, interferons. And that's not just a single example, it's a pretty robust and consistent finding you can see here. So, well, we also asked, everyone is talking about inflammation in COVID, and people are measuring interleukin-6 and TNF and all of that and the cytokines. Why not take a look at the cells in the blood and say, are those the cells? Is it indeed the blood cells themselves that are producing TNF and interleukin-6, which we did in a fashion similar to the previous experiment, basically taking blood cells from a healthy person, shown here in this column, stimulating these blood cells in tissue culture with a cocktail of stimuli. We call this the bacterial cocktail. So it is as if those blood cells have seen some kind of a bacterial infection. And you can see that there's a very robust production of TNF and interleukin-6. However, in a COVID infected patient, shown here on the column on the right, this is severely impaired. So again, even though we had heard on the news and there were papers coming out, there's lots of interleukin-6 and TNF in the blood, we learned from this experiment, wherever those molecules, cytokines, were coming from, were not from blood cells. They must be coming from somewhere else in the body. Similarly, we stimulated these cells with viral cocktail, again, similar results. So all in all, this led to the notion that unexpectedly, the cells in the blood were in a state of hibernation, if you will. They were unable to do their job. And this was consistent with other things like NF-kappa B activation was diminished in these dendritic cells in the blood. Okay, so I did mention cytokines. People were measuring interleukin-6 and many, many other cytokines. And consistent with those results, we also saw high levels of interleukin-6 in the plasma, TNF-alpha in the plasma, and MCP3 in the plasma. And interestingly, patients who were critically ill in ICU, shown here in blue, had very high levels. Patients who were severe had a bit lower levels. Moderate patients had low, and then the healthy patients, that's essentially a background baseline level. So there was the levels of interleukin-6 and TNF and MCP3 were critically dependent on disease or correlated with disease severity. What we also discovered, which I'll come back to towards the end of this presentation, were 3 molecules that people had not seen before: oncostatin M, TNF superfamily 14, also known as LIGHT, and another molecule called NRAGE. These were also highly expressed in COVID patients and really tightly correlated with disease severity. As I said, these had not been seen before, but to us, this was a very significant observation because these molecules had been very well described to be involved in inflammation in the context of other kinds of lung inflammation, like COPD, asthma, allergy, and so on. So the fact that we were picking these molecules up in COVID patients, and more particularly, they were associated so strongly with disease severity, was quite striking to us. So to get a deeper handle on this, we did what's called site-seek. This is a fancy name. It's just a fancy technique where you can probe every single cell and probe every 30,000, all 30,000 genes in its genome and see which one of these genes are switched on, which one of these genes are switched off. It's called site-seek. So we analyzed 8 COVID subjects, 2 of which were convalescent, 5 healthy subjects, and 2 influenza-infected subjects and 2 RSV-infected subjects using this technique called site-seek. And here's a beautiful looking UMAP. Now we analyzed 84,000 single cell. Each dot is a single cell. And you can see in a drop of blood, you can see all the different types of cells that are present in a drop of blood. It's fascinating. These are all the cells. There isn't any other cell that you haven't captured. So it's like a kaleidoscope of cells. And what was immediately obvious when we looked at COVID-19 patients versus flu patients versus RSV versus healthy, that the gene expression profiles were dramatically different. You see the healthy patients are shown here in gray, obscured by some of the purple that's overlaying it. But by and large, they occupied very different spaces as, in this UMAP. And interestingly, if you looked at severe COVID patients versus moderate COVID patients, again, there's a very distinctive transcriptional or gene signature of a severe COVID patient shown here in orange that lies quite different and it occupies a different space from where the moderate patients are, or from where the convalescent patients are, shown here in green. Now, I mentioned interferons, which are the cytokines that I told you is important in defending against viruses. And I also mentioned that we felt that in COVID patients, interferon response was crippled because the cell type that produces interferons were unable to produce interferons in COVID infected patients. So we said, is that really so? Should we look at these 84,000 cells one by one and ask, do any of them produce any interferon? We could find in this vast sea of gray, there's not one speck of red. Not a single cell out of 84,000 cells produce interferon alpha or interferon beta, its sister cytokine. However, we were surprised by the redness we saw with these IFI27, IFIT-M3, which are molecules that are switched on by interferons. It was, if you will, the footprints of interferon response. So how can there be footprints of an interferon response when there was apparently no interferon in the premises? This was a puzzle. So, and we dived into this footprint of this interferon response more closely. We said, let's take a look at one of these cell clusters, call it CDC2. It's just a cell type that we picked up, and said, let's look at this interferon footprint. Now, these are all the interferon footprint genes. That tells you at some point there had to have been interferon. So as we said, which of these patients we're looking at have this interferon footprint? So this patient, COV5, COV6, COV7, they have a lot of red. So that's a lot of interferon footprint. Who are these patients? These are moderate patients, all right? COV2, the red patients, COV1, COV3, who are those patients? Those are severe patients. Whoa, those don't have much. Maybe they have a trickle here, but not nearly as much as the purple patients. Now, what about these black? So those are the healthy donors, 1 healthy donors, 4, 5, there's nothing there. And these are the convalescent, there's nothing. So this said to us, perhaps this interferon footprint is produced in moderate COVID patients, but not really in severe patients or in the healthy donors. And we looked at this in another cell cluster, PDC, the plasmacytoid DC, essentially similar results. You can see the pattern of colors are effectively the same. So we were very skeptical. We said, we're making some sort of an extraordinary claim. This requires extraordinary evidence. So now can we do not just single cell, but can we do bulk RNA-seq? So we looked at bulk RNA-seq in 18 COVID infected patients, look at all the RNA expression profile in all the cells. And what we saw was exactly what we had seen before. Indeed, there was this interferon footprint in these cells, but the added insight that came here was that this footprint was mostly induced early on. You see, this is days post-symptom onset. The lighter green is 5 days, 10 days, and then you get to 20 days. And you see generally patients who are within the first 5 days or so, this interferon footprint lights up, okay? And then we did some linear regression analysis to see what of the covariates would explain this interferon footprint. Is it age? No. Is it disease severity? Somewhat. Is it time since post-infection? Yup, that's a pretty strong variable there. Is it gender? No. And something else that's residual. So now we know that the disease severity and time post-onset, symptom onset, was critical variables in this footprint of this interferon signature. And so finally we said, okay, we said we didn't detect any interferon, but can we now use a very special technique that can detect femtogram per mil quantities of interferon alpha in COVID patients? And that's what we did. And you can see there's this beautiful kinetics. So this is 0 days post-symptom onset. This is about 5 to 8 days post-symptom onset. There's this initial spike, but look at the levels, femtogram per mil. That is very low, okay? Unless you had the sensitive technique, you wouldn't detect it. But look how it comes back down. So you see, what we think is that there's this very transient but blunted interferon signature in these COVID patients. So we correlated that quantity that we're measuring by this technique, this highly sensitive technique, with our interferon footprint gene signature. And you see this beautiful correlation there. And this is now consistent with some other papers. So this paper from the group in France, in the Pasteur Institute, shows that there's this early rapid wave of interferon that then is suppressed in severe disease. Then there was a paper that was published last week in Nature Medicine from the United Kingdom, where they show interferon alpha, again, using the similar technique, femtogram per mil amount. Look, it just, this early spike, and then it goes away. And then another paper that shows the same thing, and then another paper. And then, but there's one paper from the Yale group where apparently we see some different results. So that needs to be resolved. This is an important issue given that interferon therapy is being considered as a potential therapy for COVID patients. So finally, what we also saw was that diminished HLA-DR expression in myeloid disease and monocytes. HLA-DR is just a molecule that's involved in antigen presentation. We were able to confirm this with CyTOF, and I mentioned NRAGE, which is one of these molecules that we picked up that we think is involved in inflammation by cytokines in the plasma. It turns out that that NRAGE molecule is expressed in these monocyte subsets, and the more severe you are, the lower of HLA-DR you express, and the more of this NRAGE you express. So that's fascinating. The final piece is that we wondered how on earth is all this happening? And of course, we knew that the lung is not a sterile environment, because in the upper respiratory tract, you have lots of bacteria, you have our own microbiome. And we thought, could that play into this? How does it play into this? So we did a very simple experiment to measure endotoxin in the plasma and bacterial DNA in the plasma. And you can see in severe patients or critically ill patients, there's lots of endotoxin and bacterial DNA compared to moderate and healthy patients. And amazingly, if you correlate the amount of bacterial products, in this case DNA, the same thing also happens with endotoxin versus interleukin-6, TNF, MCP3, and all these other cytokines, there's a beautiful correlation arguing that there is indeed a role for this bacterial products in augmenting this inflammation that you're seeing in the lung. So this summarizes our findings in this paper. I think what this paper, the results are suggesting, is that the pathogenesis of this infection occurs, if you will, early on, where you have mild disease. There's this early wave of transient type 1 interferon production at very low quantities. This we think is coming from the lungs. This early but blunted wave of type 1 interferon leaves a footprint of interferon signature that we were able to pick up using our transcriptional analysis in the blood. That footprint was only apparent within the first 8 days or 10 days, but then it went away because the interferon went away, no interferon, no footprint. This then led to this the later stage of disease, severe disease with excessive inflammation, lots of inflammatory cytokines, lots of bacterial products. And surprisingly, what we saw was that while there was a raging inflammation in the lung, in fact, elsewhere in the blood, the immune system seems to be in a state of hibernation. By that, I mean the cells that are capable of fighting a viral infection are unable to produce the very cytokine that gives them their power, interferons. And monocytes and other cells seem suppressed. We also saw these suppressor cells called myeloid suppressor cells. So let's step back from the details and ask ourselves, what can we learn? Is there some general principle that could be suggested by these data? So I'd like to propose this principle: spatial dichotomy of the immune system. Spatial dichotomy, because the lung is an immune organ, the blood is another immune organ. They're separated by space. What's going on in the lung is raging inflammation. What's going on in the blood is suppression of innate immunity. So two dichotomous immunological events in response to a single virus. Is there some interconnection between the two? And so what we've seen with COVID, to what extent does this apply in other kinds of tissue infections? For example, severe influenza or other kinds of respiratory infections or other kinds of other tissues. This is a general principle in immunology. We're investigating this and we're looking at this factor X or factor X's that might mediate these communications. So finally, just a couple of slides. I mentioned this whole thing about these molecules we had found in the plasma that were correlated with disease severity. One of the molecules was this TNF superfamily 14, also known as light. In fact, it turns out that this light has been previously shown to be critically involved in inflammation in autoimmune disease or in lung inflammation. It binds to two receptors, lymphotoxin beta receptor and the herpes virus entry receptor. It turns out that Biogen, this company in Boston, have a GMP quality reagent that is capable of blocking the interaction of light with its receptors. Essentially, it's a lymphotoxin beta receptor fusion protein. It's also called Barmilacept. It's been tried in Sjogren's disease in clinical trials and in rheumatoid arthritis patients with, unfortunately, it failed in those clinical trials. But effectively what it does is to prevent the interaction of light with either one of its two receptors. So we reached out to Biogen, and so we reached out because of our results, but also as we looked at the literature based on our results, we were amazed to see that there was a paper from Rafi Ahmed's group that was published 20 years ago in Nature Medicine, in which they had used a blocking antibody against lymphotoxin beta, effectively the same kind of antibody as this reagent from Biogen, and shown that in fact, if you blocked the interaction of lymphotoxin beta receptor with its ligands, you would ameliorate the symptoms of respiratory failure in response to LCMV infection in mice. So this is a control antibody, all the mice die. If you use that blocking antibody, the mice then survive. If you block TNF, you don't have any benefit. And if you combine anti-TNF antibody with this lymphotoxin beta receptor, you don't have any benefit. So that's prophylactically. And this works even if you give it therapeutically 3 days after infection, you still have this tremendous, some effect on survival. So that's the experiment we're doing. We signed a deal with Biogen. We're doing an experiment in hamster to test the efficacy of their reagent in preventing lung disease with COVID-19. This experiment is starting next week. We'll have the results in a couple of weeks, and hopefully we'll see. I mean, these things we can never predict, but hopefully if it shows positive results, we will do a human clinical study with Biogen. So that's what I have to say. This is our COVID-19 Task Force 1. I mentioned all the people who were we're very lucky to have working with us. And so thank you very much.
Dr. Pulendran, thank you for that. And now we'll switch over to Dr. Wang.
Yep.
Thank you. Thank you so much, Errol, and thank you, Bali. That was a beautiful presentation on the innate response and dysregulation of innate immunity in some groups of COVID patients. So in contrast, I'm going to focus on the adaptive response and specifically on antibodies that are elicited in SARS-CoV-2 infection. I'm going to talk about a specific type of antibody, IgG antibodies. These are a type of antibody that we think of as being really central in protecting us from viral pathogens. Importantly, IgGs have this Y-shaped structure, and this is comprised of two different domains that are both required for their function. This Fab domain binds to the antigen, or in our case, a viral particle. The Fc mediates interactions with effector cells at the site of infection. So cells that are expressing what are called Fc receptors can be engaged by the Fc, and depending on those interactions, the degree of inflammation will be changed and the cellular functions will be determined. Some things we know from literature that has emerged over the last several months about the IgG response after SARS-CoV-2 infection is that the vast majority of people indeed do seroconvert to produce IgG antibody about 12 to 14 days after symptom onset. We know that neutralizing antibodies are generated after a majority of symptomatic infections. And A body of evidence has emerged also suggesting that serum IgG titers do wane over the course of some weeks after a primary infection. And I thought I would just talk about this for a moment because it's a topic that's gotten a lot of attention in the media. And I think it's good to go over what the meaning of this might be. This was reported in a paper that got a lot of press in Nature Medicine in August, showing that between two time points that were about 8 weeks apart, acute to convalescence in COVID patients, or sorry, rather patients infected with SARS-CoV-2 who either had asymptomatic infection or COVID-19. Over the course of these 8 weeks, there was an observable decline in CoV-2 specific IgG levels and also in neutralizing titer. And this has been demonstrated in a few studies now. I'm just showing some other examples here. The cumulative evidence seems to support this observable decline in neutralizing antibody titer over the weeks following a primary SARS-CoV-2 infection. What is the significance of this? Do we need to be concerned about this? And I think the answer to that question is it depends on the goal. So is the goal to have sterilizing immunity for long-term after a primary infection? If so, then this might be concerning. The reason is because serum antibody is your frontline defense against an invading pathogen. That's because it's already there. It's ready to, neutralize a virus, for example. The body doesn't have to respond to an infection in order to produce that antibody. It's the frontline defense. In this setting, however, in a re-exposure to a virus, in the event that there is no sterilizing immunity, which, let's say, if, the serum antibody levels do decline to a point where some people aren't protected upon a re-exposure. The beautiful thing about the adaptive system and the hallmark of this system is that there's a memory response to infection. And it's really the quality of that memory response that will determine the outcome of subsequent SARS-CoV-2 infections. And this is going to be the focus of a lot of studies over the next year. How is this memory response established and how does it respond during re-exposure to SARS-CoV-2 infections? So I think this issue of serum antibody titers waning is not necessarily something that we have to be terribly concerned about, because in the absence of sterilizing immunity, there may still be a very robust memory response that will protect people, not against infection, but likely against disease, and we hope certainly against severe disease in many people. What I'd like to focus on a little bit here is a discussion of antibody quality. We hear a lot about antibody titers, neutralizing antibody titers, but I think equally important is the quality of the response. And let me tell you what I mean by this. The quality of an antibody response is determined in large part by its ability to signal to innate cells. And these are cells that express the receptors for that Fc domain, and they're called Fc gamma receptors, and then they have different names, 2A, 2B, 3A. At any rate, these are the receptors that are triggered by the formation of immune complexes. And so this is a sort of critical point in signaling by antibodies. We have a tremendous amount of IgG floating around in our blood, 10 to 12 milligrams per mil in most healthy adults. This monomeric IgG doesn't have a strong enough affinity for the receptors on its own to signal. However, when we come in contact with a pathogen, SARS-CoV-2, for example, IgG antibodies that can bind to the virus form what's called an immune complex. This multivalent complex now changes things altogether. This is now a signaling complex, and it can interact with cells to modulate the inflammatory response. So what determines the outcome of these interactions between the immune complex and the receptors on immune cells? This is the composition of the antibodies within the complex. So There are two determinants of these interactions. One is the protein sequence of the FC. So this is called the IgG subclass. In humans, these are called IgG 1, 2, 3, or 4. A second determinant of the interactions is the composition of the glycoform that's present on the FC. Those are the two determinants of the structure of the FC, which dictates the receptors that will be engaged by an immune complex. So if you have a complex, for example, that's comprised largely of IgG1 or IgG3 subclasses, or that have more pro-inflammatory glycan modifications, these will signal through activating receptors. Two of the most important activating receptors are Fc gamma 2A and 3A. These receptors trigger pro-inflammatory responses, cellular activation, pro-inflammatory cytokine production, some functions like antibody-dependent cell cytotoxicity, ADCC, phagocytosis. All of these are required for protection in an infection. And then, as we know, of course, inflammation is a spectrum, and an overexuberant response can lead to pathology. So on the flip side of these pro-inflammatory complexes, you can have complexes that are quite different in nature. So composed more of IgG2 antibodies or antibodies that are modified by glycans that have sialic acids. These have more affinity for this inhibitory receptor called 2B. This receptor really puts the brakes on those activating receptors. So we think of it as being inhibitory or anti-inflammatory. This difference in the composition, the difference that the composition of the immune complex makes is critical. And I want to make a point here, which is that in terms of antiviral immunity, these interactions with different receptors determine the activity of neutralizing antibodies in vivo. In vitro, neutralizing antibody titers are measured in a simple assay where you're looking at blocking of infection of cells, typically. That's often the way it's done. And that doesn't take into account these interactions with receptors on immune cells. But in vivo, in an animal or in a person, these interactions with different receptors are actually critical for the protective capacity and potency of neutralizing antibodies. I want to show you an example of this. So in a model, for example, of influenza virus infection, where mice are infected with an H1N1 virus, and then they're transferred a broadly neutralizing monoclonal antibody. In this example, it's a monoclonal antibody called 6F12, potent, broadly neutralizing antibody. This monoclonal antibody can be engineered to engage different receptors. And what you need to know here is that the mouse IgG2A is the only one of these that engages activating receptors with high affinity. And so the mice that receive the IgG2A, broadly neutralizing antibody, lose very little weight after challenge, and they're 100% protected. Whereas the mice that receive the exact same antibody, but with a different Fc domain, meaning it engages different receptors, are not protected at all. This same data is seen with, has been now demonstrated with a whole panel of broadly neutralizing influenza antibodies, and quite interestingly, also antibodies that don't have neutralizing activity. These can be protective in vivo as well if they engage the right receptors. Now, as you know, Bali has discussed, and I mentioned, of course, inflammation, the inflammatory response falls on a spectrum from being protective to potentially being pathogenic. And we can observe this in the lab in a model of chronic immune complex exposure. For example, a model of spontaneous autoantibody production where immune complexes are formed. And one of the sequelae of that is autoimmune glomerulonephritis. If you, measure proteinuria in these animals, and these are animals that spontaneously develop the immune complexes, but they either have activating Fc gamma receptors or are deleted for their activating receptors. You see that those without any activating receptors have quite delayed onset and reduced incidence of proteinuria. without that activating receptor activity. And in terms of survival, the mice that don't have activating Fc gamma receptors have much, much better survival. So these same receptors in the context of infection and protection by neutralizing antibodies can be quite important for mediating the potency of protective antibodies. However, too much of the activating response can of course result in disease. So just a couple conclusions from this. The ability of antibodies to protect often depends on their interactions with receptors, Fc gamma receptors. And inflammatory cell activation induced by these interactions can either be protective or can contribute to disease. One of the very interesting observations that I'm interested in, that our lab is studying, has emerged over the last several years from studies of human antibodies. And what we've noted is that people produce very different repertoires of antibodies with respect to their ability to engage different types of Fc gamma receptors. So some people produce antibody repertoires that are more likely to be anti-inflammatory or inhibitory, whereas other people produce repertoires that are highly activating. They have, a subclass distribution that's skewed more towards antibodies that with higher affinity for activating receptors, and glycan modifications that also increase those interactions. So how does this heterogeneity impact susceptibility to infections? This is one of the questions that my lab is really interested in. And to try and address this in the context of COVID-19, we studied samples from patients with different degrees of disease severity. So here I'm showing you summary data from patients who are PCR positive for SARS-CoV-2, but who had different disease severity. So were either hospitalized, treated in the ICU or on the floor, or with relatively mild disease who were treated as outpatients. So the length of each slice here indicates the relative abundance of each factor that's shown on the perimeter. And I'll just sort of walk you through this. There's a very clear difference here that you can see between the hospitalized COVID-19 patients and the outpatients, just on the basis of patterns of the abundance of these different types of antibodies. We observed that the hospitalized patients have higher levels of IgM and IgA relative to outpatients. In our cohort, we did not see an increase in IgG in hospitalized patients, although some other studies have observed higher IgG levels. We did not see a significant difference in the subclass distribution of IgGs. But then when we hone in on the IgG modifications, there's a very pronounced enhancement in one particular glycan. So these are in the red here, pink, showing 4 different glycan modifications, which, as I mentioned, also change the interactions with the different Fc gamma receptors. And we see that patients who are hospitalized have this enhancement of one particular glycan. We're calling this F0N0. It lacks a core fucose. So we call it afucosylated. This is a modification that's been fairly well studied. It has a very specific activity. And that is that it increases affinity for one specific activating receptor, Fc gamma R3A, which is also known as CD16A. This receptor increases cellular activation, pro-inflammatory cytokine production. It's a major receptor responsible for antibody-dependent cell cytotoxicity, and it also has a role in phagocytosis. This pro-inflammatory activity of afucosylated antibodies has been utilized in the context of tumor immunotherapies. So there's a afucosylated version of a CD20 antibody, for example, that's used in some contexts. The idea being that promoting inflammation in the relatively immunosuppressed context of a tumor can enhance clearance of the tumor cells. But in this case, we're seeing this profile in patients with more severe COVID infection. So we asked the question then, are these differences, do they create an observable difference in binding to this receptor? So we created immune complexes with IgG from patients with different levels of afucosylation on their SARS-specific antibody. And as you can see, there is a significant correlation between binding to the receptor and the amount of fucose in the immune complex, with more afucosylation correlating with greater binding to CD16, as you would expect. When we do the same thing by incubating immune complexes with cells that express CD16A or NK cells, we see that these are actually levels that do impact the activation of these cells, so that there's more NK cell degranulation observed in the presence of immune complexes with higher levels of afucosylation. So we have this distinctive antibody fingerprint in patients with more severe disease, and the level of increase in this one particular glycan does seem to be physiologically relevant in that it can modulate the activation of cells expressing this relevant receptor. So how is this regulated? This is an important question. Is this being triggered upon viral infection? That's the first question that we asked. And in order to address this, we studied samples from longitudinal samples where we had either a sample taken at the acute time point and then a later time point, either 2 weeks, 3 weeks, or 4 weeks later. The idea being that as antibody turns over, we might see a decline in the abundance of this modification if there was an acute increase upon infection. And we didn't see any significant difference over time in the abundance here. So we tried to look at this in another way, which is to ask the question, does the abundance of this modification correlate with the amount of virus present? And the readout that we used there was the CT value, so the amount of viral RNA. And again, we saw no correlation between the abundance of this sugar modification and the amount of viral RNA. We then tried another way to ask this question, which is if we look in a separate pulmonary RNA virus infection where we have a more controlled sample set, do we see that symptomatic infection makes a difference? And this was with influenza A virus. So these are samples from a controlled influenza A challenge study where we have pre-infection time points. So this first time point is a pre-infection. Each line represents one person and each dot is a different time point. And so what we see here is that infection, which took place between these first 2 points, did not significantly change the abundance of this modification in the context of influenza virus infection. So a separate RNA, respiratory virus. This is obviously not definitive data to answer the question, did infection trigger this change in some patients? But so far we don't have any data supporting that notion. So other things that have been reported and observed to change the distribution of antibody subclasses or glycoforms are age and also sex. So we looked at those 2 parameters. Age, I'm not showing the data here, we saw no correlation with age. Very interestingly, however, we did see a correlation with sex in 2 different cohorts. So in hospitalized patients from Stanford Hospital or from Kaiser hospitals, men had elevated levels of this glycoform modification over women. And so a few conclusions from these data. The quality of antibodies produced by hospitalized COVID-19 patients is distinct from outpatients, in our cohorts at least. Hospitalized COVID-19 patients have a distinct antibody signature, which is characterized in part by this modification, this afucosylated glycan modification. The levels of this glycan didn't change over 4 weeks after infection, and they did not correlate with viral RNA levels. So we don't have any evidence that this is triggered by infection itself, but we're doing much more work to look at this now. And finally, male COVID-19 patients had a higher level of afucosylated glycoforms compared to females. So some questions that we have in progress, just 2 that are sort of central to the way that we're thinking about this. An important question here is this pathway, Fc gamma R3A or CD16, is this actually contributing to a more inflammatory response that we know is driving pathology in severe COVID? Does this have any role there? Or is this a simple biomarker for patients with more severe disease, which is also important? And then are high basal levels of this glycoform a biomarker for risk? So is it possible that people who go on to develop more severe COVID-19 have this change in their antibody repertoire prior to infection? That would be very interesting and helpful as far as assessing risk, triaging potentially. So these are two questions that we're very interested in and focused on right now in the lab. And I'll just say that all of the COVID work in my lab has been run by a few people. Shaborni Chakraborty has done really a lot of the bulk of the work on this and has been excellent at running this project. Carly Edwards as well, Joseph Gonzalez and Anthony Bozenko have been sort of the core team in my lab. And then there were a number of critical and very generous collaborators at Stanford in pediatrics, in infectious diseases, in pathology, really wonderful and generous collaborators. And then I have a number of collaborators from outside of Stanford at NIH, at Kaiser, at Mount Sinai, Cornell, and University of Chicago. Thank you so much. I hope I'm not too much over time.
No, that was excellent, Dr. Wang. Thank you so much for your presentation and Dr. Pulendran's presentation. A lot of great content was shared, and it seems like we're learning so much. So first off, thank you for all the work you guys do in helping us understand this much better and make us feel less anxious about the future. We certainly have, as expected, a number of questions. Dr. Pulendran, thank you for starting to answer questions in the background. We'll give it a few minutes. I know we're going over, if that's okay with you both. And then any additional questions I can email to you if you want to still answer, you're welcome to, and we'll email this out to everybody. I'll start with Dr. Ash has a question here. Can you comment on the role of bradykinin in the immune response? A recent model posited a central role. Bali, can you turn you're on mute?
Sure, sorry, yeah. Yes, it is a very good question, and there are some data emerging on the role of this bradykinin pathway in accumulation of fluids in the lung, and it's how the ACE2 receptor might interact with it. We are now doing a study with a much larger number of patients, and we can see that we can cluster disease severity. The most severe patients fall into two clusters, and we believe that this bradykinin pathway and some of the clotting pathways seem to be defining one cluster, and the other clusters seems not to have that. We're probing much more into this and what's going on there, but it's still very early days, and there are some hints coming through that this pathway is critically important in disease severity. So, and it seems to be expressed in a subset of the severe patients, as far as we can tell.
Excellent. Dr. Wang, there's a question asking about, did you observe any differences in fecalization based on ethnicity?
That's an excellent question. We didn't have the numbers to do that in this study, but that has been observed in other studies in other contexts, not in the context of COVID. And I think it's something really important to investigate here.
Great, thank you. A question that's been upvoted a few times, Dr. Anuja Sath asks, several retrospective papers do not report an increased risk of severe COVID-19 in immunosuppressed patients on biologics for underlying immune conditions, including anti-CD20. Is this reflective of redundancies or crosstalk in innate B T responses to COVID-19?
That's a great question, fascinating. I'm afraid that I haven't followed that literature. I should. It'll be fascinating. You know, I'd love to be able to look at those papers to see what the outcome measurements were in terms of which aspect of COVID pathogenesis was perturbed by immunosuppression of any kind. I mean, were the readouts broadly just clinical severity? Were there some aspects that were more perturbed than clinical? I can't, it's a fantastic question. I'd love to be able to answer it, but I'm lacking some context to give any informed answer to that question. You know?
Understandable, understandable. We have a number of more questions. We won't get to all of them today, but I will email them out to you all. I want to perhaps, it's 9:06, I want to start to close it down, but I do want to end on a good question came from our division chief, Dr. Upi Singh, and Upi, Dr. Singh, feel free to comment if you like, but she asked about the question of all this great work you're doing. Does it give us any information about our long haulers, the patients with symptoms for many months out? Any better understanding of why certain patients have symptoms for so long?
Taia, you want to have a go at that?
I think it's an excellent question. This is something that needs to be studied specifically moving forward. This cohort on its own. You know, one of the critical pieces of information that we need now, and that studies are starting to come out, are the autopsy studies from our pathology colleagues. These are going to give us a lot of critical information on exactly what is going on in the lung, what cell types are there, what are the pathways that leading to damage. And it may be that people who have a longer disease course have something completely different going on than those who have a poor outcome early on. So I think that those studies are going to be really critical to understanding the kinetics of this disease and severe outcomes.
Yeah, Upi, thank you. That's a wonderful question. And of course, it highlights something fascinating about the pathogenesis of viruses. Why is it that some viruses cause severe disease and inflammation and death? Other viruses cause mild symptoms. Other viruses are asymptomatic, and yet other viruses are long haulers, as you put it. So these are different states of the immune system. And whilst there's a lot of effort in understanding the pathways that lead to death and severe disease, the pathways that lead to coexistence of the virus with the host in a state of truce remains kind of a mystery, or the pathways that dictate asymptomatism is a mystery. So we would love to be able to investigate this. And if you have samples from long haulers, I'd love to talk with you and actually work with you on that. So.
Yeah, the only comment I make is when you listen to the symptoms that long haulers have,
Yeah.
to me, it seems like autoimmunity, right? Because they have joint pains, they have sleep perturbations, they have all sorts of things. It's not, some will have pulmonary issues, but it's so systemic. I think that's going to be really interesting. And I think, as with the great work that you and Taia are doing, we're finding out so much. I worry, and I think that there's a possibility that this actually will be the long-lasting impact of this disease. I'm taking away nothing from the people who are losing family members to acute illness, but if you think of the 80% of people who are who are mildly infected on the outpatient, and if X percentage of them have long-standing symptoms, that's going to be a huge morbidity. So thank you for great talks, both of you.
Thanks, Ubi.
Thank you. Dr. Rogers, any questions or comments?
Yeah, I was just going to say that I think we have the opportunity from COVID to learn a lot about other patients as well. I think critically ill patients often have a very similar cytokine profile to COVID-19, but yet I do think COVID-19 in the ICU, we see that there are these patients that are very, very inflamed for a much longer time. And so just to echo Upi's point and yours, I think that we have done a bad job in critical care of following people serially for a long time and really understanding what's different about that subset of people that are sick for really long times will be so informative, not just to COVID, but also to critical care in general. We don't follow people usually after hour 72, but what's true in COVID is probably true in critical care as well.
Thank you. Yeah. So when I couldn't agree with you more, because when we saw these results of peripheral immunosuppression, we called up some experts on sepsis and said, showed them the data and said, we don't know anything about sepsis. We don't know anything about critical care. What do you think? And they said, it's funny, in sepsis patients, we see a lot of this immunosuppression. Yeah. So, I mean, it's sort of highlights that it's beyond COVID. COVID is just a seed from which you could, there's so much more. The implications of COVID immunology are broader than just COVID,
Good points. I'm really sorry to say we have a lot of really good questions that we can't get to now, but Dr. Wang, Dr. Pulendran, we'll email them out to you if you have the time. I know you're super busy to answer these questions. We do send a follow-up email in the next day, and we can get those on the website for everybody to see along with these, this whole recording. Thank you, Dr. Wang. Thank you, Dr. Pulendran, for really wonderful talks and for your time. It was really great stuff. And of course, thank you for all the work that you're doing for us. Thank you to all our panelists for joining us as well, and to all the attendees. We have Dr. Nadeau joining us next week for vaccine updates in COVID-19. So we'll see you next week, and have a great week as well. Thank you so much. Bye-bye.